Welcome to video 13 of Genetics, the Code Broken. In this video, we're going to be looking at embryonic development and how gene sequences and gene cascades are used to develop in vertebrates and how they determine limb formation in birds and mammals. So embryonic development is a term we use when we're talking about organisms that develop from a fertilized egg all the way through to their development into a whole organism. And this is controlled very carefully through sequences and stages. And in embryonic development, these stages are cell division, cell determination, induction, differentiation, and then pattern formation. When we talk about cell division, we're talking from mitosis to form millions of cells of the adult. And then there's a point when the role of the cell is decided and cannot be reversed, and that's known as cell determination. Cells influence the development of neighboring cells by communicating, and this is when the process of cell induction, and the influence of these cells can be by contact or chemical signals. We then go into cell differentiation, where similar cells become dissimilar and specialized, and this is affected by the position of a cell and the pattern of gene expression. And then the last one is the development of spatial organization of an organism. And this is where the tissues and the organs develop within the correct position for that body and for that organism for correct development. Here we see from a zygote, we see that many of the cells are undetermined. And then as we start to form an early embryo, we see that cells become determined and they can develop into the cell that they are differentiated into, and then we can see that they're over time that we produce the whole organism and the cells and structures that they require. So for embryonic development to occur correctly, there are a series of genes that must be expressed for this to take place, and this is controlled and coordinated when genes are switched on and off during this development. The genes that are responsible for this process can either be structural genes or regulatory genes. When we refer to structural genes, we're talking about genes that code for proteins involved in the structure or function of a cell. And then those regulatory genes, these control the expression of structural genes, and they usually inhibit expression until the time it is required. Due to the Human Genome Project, there have been thousands of non-coding genes identified, and majority of these genes are regulatory genes, which are involved in coordinating this differentiation and development. One specific sequence of genes are homeotic genes, and these control the development of the body plan in different animal species. So the homobox genes or HOX genes are regulatory genes, and these control the morphogenesis of an organism. And it codes for a protein that can bind to a specific gene sequence of DNA and then affects the transcription of that gene. This homeobox gene produces transcription factors, and these can switch to genes on and off. And by doing so, this can cause and create gene cascades through depending on where the section of DNA in which they bind to. The gene cascades, as just mentioned previously, these occur when a gene triggers both its coded product and a transcription factor, and it switches on the gene for the next step in a series. And by having these cascades, it controls the pattern and sequence of development in an organism. And these gene cascades work together as a network, which control the overall pattern of development. All animals have these HOX genes and that create this gene cascade, and these allow the pattern of the body to occur from front to back, so anterior to posterior along the body axis. In terms of limb formation, the HOX gene network plays many roles in developing the limbs of vertebrates, and one in particular is to code for the pattern of the structures along the axis of the limb, and then for the development of the digits. This regulation we can see here is important for the structure of the forelimb in humans, and together this series of genes is able to express specific genes and regulate this development. So it happens over a series of cascades where one has to form before the other can, and this happens over a period of time. Due to these series of genes that are responsible for embryonic development, we see an evidence of evolution. And so we see that homologous chromosomes, and they carry the genes for the same traits at the same position. In terms of these genes, they all play a similar role in developing the basic chemical molecules of a cell. So these mainly are the amino acids, nucleotides, sugars, and fatty acids. We can see from genes, we can see that the chemical processes of cells are also similar. And then we see that the nucleotide sequences in genes are remarkably similar in different cell types. That concludes video 13. Make sure you tune in for video 14.